Today we are going to talk about the article entitled Recent Advance in Hydrogel Application for a Teacher Engineer are Clinical Training, which focuses on hydrogel as a fundamental biomaterial in the field of tissue engineer and engineer medicine. Hydrogel are biomolecules composed of artificial and natural polymers with a quite three-dimensional structure. This structure have a unique characteristics such as a high hydrophilicity, excellent water retention capacity, biodegradability, compatibility, and robust medical property. The objective of this study is to review the various polymers used in the manufacture of hydrogel and their application in tissue repair and clinical care, right for skill and bombs to the later, as well their use for drug delivery. This scaffold is expected to play a significant role in the repair of numbered tissues in the future.
Now, the concept map of the article is organized around the central concept of hydrogels, breaking down the characteristics, their classification, the type of polymers that make them up, and their application in regenerative medicine and tissue engineering. First, we have the definition and characteristics. Hydrogels are biomolecules formed by artificial and natural polymers that have a quasi three-dimensional structure. They stand out because they are highly hydrophilic with a great capacity for swelling and water retention. In addition, they are biodegradable, biocompatible, and have solid mechanical properties, making them crucial in cell culture techniques, tissue engineering, and regenerative medicine. They can also act as space flying agents, drug delivery system, and even as 3D structure to stimulate cell differentiation. In terms of classification, hydrogels can be divided according to various criteria. For example, depending on their sources, they can be natural, synthetic, or hybrid, depending on their ionic charge. They can classify of cationic, anionic, or anonionic. They also differ according to the cross-linking method, which can be physical or chemical. By their physical properties, where we find smart or conventional hydrogels, and according to their responsiveness, which can be chemical, biochemical, or physical. Finally, they are also classified by their preparation methods, such as copolymers, homopolymers, or interpenetrating polymers. Other criteria include their swelling nature, degradability, and crystalline structure. The article also discusses the polymers used to manufacture hydrogels, who are divided into broad categories: synthetic polymers and natural polymers. Synthetics include ethylene peroxide, a water-soluble polymer that stands out for its high ionic conductivity, solubility, and high crystallinity, thanks to its ability to form hydrogen bonds with water. It is considered an excellent option for creating hydrogels. Polyvinyl alcohol, widely used in pharmaceutical and biomedical applications, is structured with hydroxyl groups, allow physical or chemical cross-linking, and its different molecular weights influence properties such as solubility, flexibility, adhesion, and mechanical strength. And polylactic acid and biodegradable and biocompatible polyester obtained from renewable sources such as starch, highly valued in medicine for its no toxicity and versatility. As natural polymers, the following stand out: agarose, which is extracted from ocean bacteria and has advanced application in drug delivery; arginate, weakly used in regenerative and biomedical medicine due to its biocompatibility, low cost, low toxicity, and cross-linking capacity. It's key in wound healing, tissue engineering, and bioprinting. Hyaluronic acid, present in extracellular matrix, important for joint lubrication and tissue homeostasis, with application in tissue engineering, drug delivery, and viscose supplementation. Finally, collagen and gelatin, which are biocompatible, biodegradable, and low antigenic polymers. Collagen is most abundant in protein in the body, while gelatin is obtained from its hydrolysis but are widely used to as in bionics and tissue engineering application. Regarding the applications of hydrogels in tissue engineering and critical trials, the article highlights several very important fields. First is wound healing. Hydrogels are very useful because they help maintain moisture in the affected area, prevent water loss, promote cell proliferation, and facilitate nutrient exchange. In addition, they have antibacterial properties that promote recovery. Some examples are chitosan combined with agarose or PVA. Ongoing clinical trials for the treatment of ulcers, dermatitis, and burns are even mentioned. Another application in bone regeneration because hydrogels are highly hydrated polymers, they can replace bone materials. When combined with ceramic face, their bioactivity and osteoconductivity increase. Polymers such as collagen, chitosan, and gelatin are particularly attractive in this field. In fact, there are clinical trials focused on the treatment of knee osteoarthritis. We also find joint therapy in synovial joints where there is a high level of friction and where idle health, such as PVA, can mimic the lubricating and mechanical properties of natural cartilage, making them a promising option in cases of osteoarthritis. A specific example is the gel sticks implant, which is currently in phase 3 clinical trials. In the case of the liver, hydrogels play a fundamental role in liver tissue engineering as, the, as they allow the creation of implantable structures in vitro models and by artificial liver device. Thanks to this, they help maintain and preserve the functions of cells that depends on anchoring surfaces. Finally, another very notable application is drug delivery systems. Hydrogels function as smart systems capable of releasing drugs in a localized and controlled manner. Their great capacity to return water without dissolving, together with their response to different environmental stimuli, makes them ideal for improving the effectiveness of treatments while reducing side effects. Finally, we move on to the final analysis and future considerations regarding the scope of hydrogens. These materials represent a truly revolutionary technology in the field of tissue engineering and regenerative medicine, thanks to properties such as their biocompatibility, biodegradability, hydrophilicity, and ability to mimic the biological environment. The article shows how both synthetic and natural hydrogens are being successfully applied in different areas, from wound healing and bone regeneration to joint therapy and drug delivery systems. In addition, it highlights the clinical importance of the topic by including a detailed review of specific polymers, along with various clinical trials that demonstrate their tangible progress. However, certain challenges are also pointed out. For example, some natural polymers such as alginate may contain impurities that trigger immune responses, or hyaluronic acid, which on its own needs to be reinforced for applications requiring greater strength. Similarly, although combinations such as chitosan with gelatin have shown promising results in vitro for skin tissue engineering, their in vivo applications still require improvements in mechanical strength. Looking ahead, the article proposes several lines of research and development. optimizing properties, improving the strength and stability of hydrogels through new cross-linking methods or nanocomposites. Customization and precision, creating smart hydrogels that respond to stimuli such as pH, temperature, or enzymes to release drugs more specifically or more accurately reproduce the microenvironment of certain tissues. Overcoming current limitations such as the purity of natural polymers, immunogenicity, and scalability in the production of complex hydrogels. Complex tissues, for example, in the creation of organs such as the liver, allowing cells to maintain their functions both in vitro and in vivo. Key, the development of new disease models using hydrogels to recreate 3D environments that simulate real conditions, which can aid in the research of pathologies such as cancer or neurological diseases. And advances in 3D bioprinting, where hydrogels are integrated into technologies that allow the construction of customized and functional structures with complex geometries and specific cellular compositions. In conclusion, the future of hydrogels focuses on continuous innovation, perfecting their properties, expanding their application to more complex tissues and organs, and consolidating their role in personalized